Hello guys, welcome to Big Book. Today we are going to discuss about flow cytometry. It's a technique to count, examine and sort microscopic particles suspended in a stream of fluid. Here this is a technology where we can evenly count the number of the particles which we are examining and also sort the microscopic nature of that particle, where we can understand a certain features of the particle, and also it is used in many fields of the science. As we are discussing in the biology, we are going to use it for the cell biology count. Hence we can find the multiparametric analysis of the physical and chemical characteristics of single cells with an optical and electronic detection apparatus. Here we can understand each and every cell, okay, and its size and the whatever the nature of that cell, depending upon the certain parameters which has been already placed in the computational system, that is the analogs or the detectors which is connected to the computer system, where we are going to get the results accordingly. Schematic representation of a flow cytometer showing the sheet fluid, sample and the light source, forward detectors, sideward detectors, electrical ring, and the further next slide will be showing the further schematic representation. Sheet fluid is one which will be having a uniform viscous, keeping the sample cells separated from each other, which is going to pass through the light source and also from the electrical ring, further giving the taking the charge for the each and every cell depending upon whatever the dye has been detected, or the fluorescent dye has been detected from the detectors, which are the forward scatters and the sideward scatters. Forward scatters are parallel, sideward scatters are perpendicular to the source of the light. And electrical ring is placed at the aperture, which is just below. And in this slide we are seeing that the fallout of these cells in the droplet forms having the charge gained by the electrical ring according to the dye is going to get deflected from the electromagnets, and further the charged gained cells going to get deflected and it is going to fall into the respective containers, and the containers can be two and more depending upon the number of the dyes has been applied in the sample. According to that, the sorting or separation will take place. This slide shows that how the beam of light can get scattered according to the two-dimensional or three-dimensional kind of the scattering, where from one side the beam has been passed through the droplet and the overall scattering has been shown. In the principle, a single wavelength beam of laser light is directed onto a hydrodynamically focused stream of fluid. Hydrodynamically focused stream of fluid is one which we discussed, that the sheet fluid and the sample is mixed in this flow cytometer, where sheet fluid is uniformly viscous to keep the cells which has been suspended in the form of the sample to be separated from each other, so that it can pass through the narrow aperture and the narrow base of the tube too, so that each cell passes through the light one after the other. Light detectors are aimed at the point the stream passes through the light beam. Now, detectors are placed after point of beam passes through stream fluid. Forward scatters are placed with the beam. Sideward scatters are placed perpendicular to the interaction point. So here, whatever the sample passes through the light beam, okay, if the forward scatters are the detectors, sideward scatters are also the detectors which are going to absorb the amount of the beam has been further scattered after absorbing by the sample. As the number of the detectors are present, that good the signals will be sent to the source of the computation and the results can be analyzed. At each particle the beam scatters light and fluorescence found in the particle get excited and emits light at lower frequency compared to light source. Scattered light and fluorescent light frequencies are picked and considered by the detectors and passed to the computation source, that is the computers, and further the result will be seen. Fluctuations in brightness at each detector is possible to note various types of information of physical and structure of each and every particle. So whatever the multiparametric kind of the measurements has been already decided, according to that the physical and structure of the each and every particle or the cell will be absorbed by the detector. Whatever the scatters they are going to get or absorbed as a signals, and that will be taken into the result within the computers. Flow cytometers are able to analyze thousands of particles every second in real time and can actively separate and isolate particles per the specified properties. So here flow cytometers are so efficient that they can analyze thousands of particles in every second. So it means that we can use the huge sample to analyze whatever according to the multiparametric decisions or the informations which have already put on the computers. Now before to that we have to take the solid tissues, which we have to convert it to the single cell suspensions, which is further taken into the flow cytometer apparatus, so that we can pass it to a procedure and we can analyze the particular sample. Coming to the next slide, main components of flow cytometry. Flow cell is nothing but the topmost part of the flow cytometry, where we are going to suspend the sample and the sheet fluid, which is going to pass the single cell through the light beam for sensing. The next component is the light source, which will be having lamps, high powered water cooled lasers, low power air cooled lasers, diode lasers. These are the light sources from where the light will be emitted towards the sample on the narrow tube. Next, lamps will be xenon or mercury. High powered water cooled lasers will be argon, krypton, dye lasers. Diode lasers are blue, green, red and violet lasers, which are going to be used to pass the beam through the sample at a narrow tube. In the next slide, the third component is detectors and analogs to detail conversion systems. So here it detects and generates the forward scattered lights and the sideward scattered lights and fluorescent signals and wavelength, which are going to be combinedly passed through the fourth component, amplification system, which is linear and logarithmic. Amplification system, in the sense all the signals will be respectively sent to this amplification system by the detectors. So those amplification systems will send that particular signals to the respective parameter. As this is the multiparametric analysis, the computers are going to take the signals from the amplification system and converts into the result. In this slide, modern flow cytometers with increased number of lasers and detectors allows multiple antibody labeling. So here, with many number of lasers and detectors, we can do multiparametric measurements at the efficient rate, so the antibody labeling can also be done. Certain flow cytometers can take digital images of individual cells and allows for the analysis fluorescent signal location on the surface or into the cell. So with the help of these flow cytometers, in the advanced kind, we can take the digital images of each and every individual cells, where it can also make the analysis which will be on the cell surface, and it can also be within the cell. Coming to the next slide, generated data can be plotted single dimensions, histogram, or into 2 dimensions dot plots. So regions on the plots are separated based on fluorescence intensity. So here we have given the enough amount of the information about the flow cytometers and how does it work on the basis of the principle, and also the components which has been used. And for this flow cytometer, an example is also be prepared as the video of the fluorescent activated cell sorter. So I request the viewers to watch even that video to make an example very understandable. Thank you for viewing the big book and this topic.